Obesity is a metabolic disease characterized by a chronic subclinical inflammatory response associated with an imbalance, dysregulation of cellular homeostasis in response to excessive nutrient intake and accumulation. Obesity poses severe risk of developing diseases associated with chronic inflammation, including type 2 diabetes mellitus (T2DM), cardiovascular disease (CVD), liver disease, cancer, and autoimmune disorders. Visceral adipose tissue, at omentum, mesentery, and the liver regulate the metabolic activity of the body. These organs are prone to inflammation. In white adipose tissue, there is a change in the qualitative and quantitative characteristics of immune cells and a dynamic interaction between them and adipocytes, leading to an inflammatory process, impaired metabolic homeostasis, and insulin resistance (IR). Adipocytes, macrophages, and dendritic cells (DC) play the role of antigen-presenting cells (APCs) and activate resident CD4 T-lymphocytes in adipose tissues. This review aims to analyze the factors influencing the formation of pathogenic Th17 (Th17) cells in obesity. A small number of Th17 cells are present in the normal healthy body that are involved in protective functions against pathogenic microorganisms. CD4 T lymphocytes form different populations: Th1, Th2, Th9, Th17, Th22, and Treg cells, which have phenotypic and functional differences. Pro-inflammatory Th1 cells produce IFN-gamma and play an essential role in the development of obesity. Lipotoxicity associated with obesity leads to an imbalance and change in functional properties of Treg and Th17 cells in the intestine, adipose tissue, and liver. The Th17 cell population links innate and adaptive immunity and has unique effector functions, plasticity, and self-renewal. Th17 lymphocytes are described as both key protective agents of mucous membranes and as prominent participants in the pathogenesis of many autoimmune disorders and inflammatory diseases. Th17 lymphocytes, depending on the microenvironment, can form pathogenic and non-pathogenic subpopulations. Systemic inflammation induces the reprogramming of the transcriptome of normal Th17 cells formed in epithelial tissues, which acquire new properties. A zone of overlapping state exists between IL-17A producing cells, which does not allow a clear boundary between non-pathogenic Th17 and pathogenic Th17 lymphocytes. It is assumed that in obesity, the pool of inflammatory pathogenic Th17 cells with cytotoxic potential is a fraction of terminally differentiated memory lymphocytes and is responsible for developing autoimmune reactions.